Thank you very much for the introduction and the invitation. It's lovely to be back and have 40 minutes to take you all the way from anatomy to the clinic. And at the same time, we're covering about 18 years of my life. So bear with me, it's going to be a long distance we have to cover. But I hope you enjoyed. If there's a YouTube symbol on the slides, that means that we have a dedicated YouTube channel. So for that particular slide, there's an entire talk just as background. So if there's anything that takes your fancy, you can go down the rabbit hole. Now, let's start with something obvious, but not that obvious. And there is no one type of anatomy. There's very different types of anatomy. We have surface anatomy, looking at the dura and the sulci. Sectional anatomy, we cut through and you look at the brain nuclei, the brain-wide boundaries. Connectional anatomy, that looks at the wide matter connections in the brain. And functional anatomy, so classical fMRI, globology, as some like to call it. Now, that is important because every anatomy comes with their own anatomical terms. And it's not like we don't have enough of them. So you also have to learn the equivalence between what we call, for example, the precentral gyrus in structural imaging, or VA6 in sectional anatomy, or M1 or the primary motor cortex in functional imaging. Now, as I said, this may seem very obvious, but I show you that it's not obvious in a minute. So show of hands, this is an easy question. How many lobes does the brain have? I see people counting already. Brilliant. Right, just raise your hand. Who thinks it's 1, 2, 3, 4, first hand going up, 5, few more hands, 6, people are still debating, 7, okay, some people are missing an 8 on the slide, just to let you know. But the point that I'm making is, you see that this should be a very easy question, but we do not agree. And the reason we don't agree is because the definition of what a lobe is and how many lobes the brain has changed over the centuries. So if you thought we have 4 lobes, then I'm sorry to tell you, you're stuck somewhere in the 1895s. If you thought it was 5, then you already moved to living history 1975. And if you thought it was 6, then welcome to the 90s. So the reason is that in the 1970s, the insula was defined officially as a lobe in the brain. And then in the late 90s, the limbic lobe was officially included as its own lobe. Now, there's still debate on whether or not that is actually the way it should be, but that's a different matter. So the point that I'm making here is that anatomy varies depending on how you look at it and depending on your previous knowledge. Now, because that actually coherently answered the question, I thought I asked things that are supposedly smarter than us. So I asked AI, starting with Google. In May this year, Google thought it was 4 lobes. I asked again, I'm very persistent, I asked again in September, and it learned and it updated to 6 lobes. The reason is someone published a paper in the meantime. Now then, ChatGPT is now about a year old. Here we go. I asked ChatGPT, and the first time I asked, it said 5 lobes. Then I just repeated the very same question. Now it changed to 4 lobes. And I just kept asking it again, and it said 6 lobes. So don't GPT. Even more so because this little ChatGPT interaction was in May. If you ask it now and you're cheap like I am and you use the free version, it's stuck on 4. It just keeps, no matter how many times you ask it, it says it's 4 lobes. Haven't tried the paid version if anyone wants to enlighten us if that. But the point is, AI also doesn't know really how many lobes the brain has. I also asked Google's BART, and it actually came up with 2 additional lobes. Any ideas? They also wanted 8. It counted the cerebellum as individual lobes as well. So depending on who you ask, you get very different answers. Now, that is important because it also alludes to the fact that we have different models of how the brain works, and depending on the framework that you use, you may have slightly different results. Now, one of the most famous models that you are all aware of in one shape or another is localizationism. The idea here is that these three famous cases that I'm sure are no strangers to this audience, they became famous because they suffered some kind of injury to their brain that then firmly placed a cognitive function in that part of the brain. So the first one is Phineas Gage, and you see the iron rod that he's shot through his skull, and his brain landed a couple of feet away. And legend has it that he got up, holding his eyes, saying, "I think," which is a very fair assessment of the situation. It's preserved, and it placed, issue. It also placed the localization of inhibition and personality in the frontal lobe. Now, Louis-Victor Le Bon is one of Broca's famous patients, and you can see that he suffered a lesion here to the inferior frontal gyrus, became unable to articulate full sentences, and that's why the articulation of language is located, as nowadays referred to by many as Broca's area. Now, oh gosh, it's going to happen over and over again. If you ever get a chance to meet Nina Dronkas, she's based in San Francisco at Berkeley University. Get to know the story about how she scanned her brain in Paris, driving one of the most important specimens we have from a museum to the scanner in a cab through Parisian traffic, and then wrapping it in a diaper to scan it. It's a brilliant story if you ever get the chance to talk to it. Now, the last patient here is better known as patient HM, and he's the one who had a bilateral surgery to his medial temporal lobe and became unable to form new memory, placing memory function in the temporal lobe. This is the idea of localizationism. So one part of the brain is responsible for one cognitive function. Now, recently I read a paper that went back and challenged it, embracing an anti-localizationism movement, which I do disagree with the name. But what really resonated with me in the paper is that they write, indeed, this is a centuries-old debate, but one, as we will argue, that is deserving of its renewed attention, given how historical limitations in computational and experimental methodology have shaped our current conceptual and empirical understanding of the human brain. These effects have important resounding implications for current research. So again, it makes that point about depending on the model that you have, that will alter the outcome that you have. And the reason this resonated with me is because we also published a paper last year where we tapped into a similar argument, basically going through the evolution of language through time, and showed that if we go with the localizationist or modular approach to how the brain works, where you have one area that does one function, we don't even have the computational power in the brain to start with something as complex as language. So we go through these different models that you can apply and actually conclude that the integrative model is the most likely model to facilitate something as complex as language in the brain. Now, the link to the paper that I've just shown you before is that when we published this, we had two teams. One team came out and said, "Wow, radically new, how dare you?" And the other team came out saying, "Well, what are you talking about? We knew this for a long time." And that's exactly the debate that the other paper, there's a preprint at the moment, literally writes in there. That people come out and say, well, this is nothing new versus this is a century-old debate. So you can choose where you want to fall on the spectrum. Now, in order to study the connections in the brain, which is what I do most of my time, we have only two methods available, one of which is called Klingler postmortem dissections. And the name gives it away. It's post-mortem, so not ideal if you want to repeat your analysis. It also very much limits the pool of data that you can work with. Specimens are difficult. Now, I started doing post-mortem dissections back in 2006, and I got extremely fascinated by this technique. And very luckily for me, around the same time, diffusion-weighted imaging tractography was born. So I started looking at white matter connections in post-mortem brains and in the living human brain. And here's a couple of papers where we validate the two methods using each other. Now, back in the days, we considered post-mortem dissections to be the gold standard to validate tractography. This has changed because they suffer very similar limitations in the end. I'm happy to discuss that after in more detail if you're interested. Also, if you look at the first one in 2011, it was still enough to just show the tracts in the brain to make it to Nature Neuroscience. It doesn't work anymore. Now you have to be a lot smarter than we were in 2011. So that's the sad news I have for you. Now, coming back to language in the brain, this is going to spin again, I guess. I'm sorry. So these are the three famous people that shaped our understanding of the neurobiology of language. We have Paul Broca. You've seen one of his patients. Here's the other one. Carl Wernicke came out a little bit later and said, well, I also see stroke patients, but they don't have a lesion to the frontal lobe. They have a lesion to the temporal lobe. And their behavioral presentation is such that they're completely fluent. So they don't have that hesitation that Broca patients show. But what they say makes absolutely no sense. And also their comprehension is impaired. So he proposed a new model of language in the brain where you have two areas, one for articulation and one for comprehension. And he also postulated the first model of language in the brain because he said, now very interestingly, in the original publication, he shows a right hemisphere model. He doesn't explain it. I tried to find the solution, why it's the right, if it's just if someone flipped the image and it's not supposed to be, or if there's actually some reasoning behind. But the first model of language in the brain that we have is actually a right hemisphere model. Now, there were a few more iterations along the way, but then sometime later, Norman Geschwind, the American neurologist, came about, and he postulated that we need the inferior parietal lobe, the angular gyrus in particular, to connect to those two regions to have concepts and abstraction in language. And he changed the connection between these areas from, as you can see here, is a very short direct connection. And Wernicke called it a psychic arc initially that was going through the insula. And now we're going all the way around the Sylvian fissure. And the model changed to the arcuate fasciculus, arching fasciculus, basically, as the main language tract. Now, if you put this all together, the neurobiology of language in the brain looks like this. We have three cortical areas scattered in the frontal, parietal, and temporal lobe. And we have the arcuate fasciculus that is connecting the three centers, either directly in red or indirectly in green and in yellow. Now, this is a model that you find in most textbooks these days, and I'm sure you've all come across it at some point in your career so far. And I'm going to spend the rest of my time now to convince you that this is wrong. So let's start with the cortical anatomy. Tremblay and Dick back in 2016 went to some experts in the field and asked them where they think Broca's and Wernicke's area are. The experts in the field do not agree. So you have a composition whereby Broca's area is sometimes considered area 44, 45. Sometimes it includes 47. Sometimes it's only 44. Sometimes we go posteriorly and include the ventral premotor area. And there's a couple of more iterations of the scenario, what we consider as Broca's area. So when you read any papers, dig deeper, see what they actually mean by Broca's and Wernicke's area, because the agreement in the literature for Broca's area is less than 50%, far from ideal. If we're looking at Wernicke's area in the temporal lobe, it's even less than 30%. So that's a big problem that we have in the literature. What you see here is a model of how vision and auditory processing works in the brain. And every box that you see is a cortical area, and every line is a connection between those cortical areas. Now, if we accept that something as simple, if you want to call it that, the visual system and the auditory system are so complex in how it's processed in the brain, then surely when we look at language, this model probably doesn't quite fit the bill. Now, there's a couple of reasons we have these stark differences. And the most obvious one is that the first two models come from comparative studies. So they were conducted mainly in monkeys, and can't do it for language. Likewise, the way you trace connections in animal brains is very different to how you can trace them in the human brain. So we can't apply the same methodology. And that's why our model of language is still somewhat underrepresenting the truth. Now, with the help of tractography and also using other modalities like direct cortical stimulation during surgery, the classical language model has been expanded. So we started in the 19th century with just that direct connection between frontal and temporal areas. Then in the early 2000s, indirect connection to the inferior parietal lobe was described. And since then, all of these connections that you see on the right have been implicated for language sciences. Now, that's the model. Another problem that I have with this model. Know who do you dare to speak up and tell everyone? You dare.
Second on the left is Rita Levi-Montalcini.
Mm-hmm. And who is that? What's that?
You can say that.
Nobel, it's not a trick question, it's a Nobel Prize question, yeah. So what you see here is correct. See, now you made a start, now they're coming. So what you see here is all the women who won the Nobel Prize for medicine. As 13, that's as many as there are. Now I asked my students with the average face of a female Nobel laureate, so we know what we need to look like to win the Nobel Prize. And this, sorry, their names first, is the face of the average female Nobel laureate winner. Now, a couple of things that are important here. Number one, the more individual you are, the better. So if I can point your attention right here, leopard print, way to go. Anyone can recognize you if you wear leopard print. Here we go. The other thing that is important is that even though the face in the middle does look like a face, it has all the facial features that you would expect, it doesn't look like any of the individual faces around it. And likewise, none of the individual faces around it actually look like the average image in the center. Now, the reason I'm making this point is because it is plain obvious when you look at faces. It's a lot less obvious when you look at the brain. This is now the average 152 MNI brain, 152 meaning that we average people, and this is what the average brain looks like. You don't see that difference that we just saw in the faces. Now, what you see, however, is that there's areas where we have a lot of agreement between people, so this is where you get sharp boundaries. and areas where you have less agreement. So this is where the template becomes a bit more blurry. And just to give you two examples, one from the gray matter. So here's the motor cortex that classically, when you try to identify it, you look at this inverted omega shape to identify the hand knob area, but it could be anything between an epsilon and a straight line. Just to show you how variable cortical area can be. Now, when we look at the connection anatomy, it doesn't get better. So what we see here, this is healthy people, by the way. If we look here, the uncinate fasciculus in pale blue here, it's barely visible in this student, and it's extremely prominent in this student. You can do this for all the tracts, and you will find there's quite a degree of variability between us. Now, this is obviously hand-selected examples, but we did look at this on the group level and actually did a variability study. And here I can show you the maps for the gray matter and the white matter, where you see the areas of variability in greater color. So the warmer the color, the higher the degree of variability. And what you can see is that in both cases, we tend to have a gradient, whereby areas that are deeper in the brain and older tend to be more alike between us. And as you go more lateral, there's more variability. This is extremely important, not just because it's a very pretty picture, I think, but also because we tend to use atlases a lot in neuroimaging. And I'm going to, this is tricky. So if you use an atlas and you look at areas where we are alike, that's fine. But the second you look at areas where we're variable, the atlas will fail. And particularly in a clinical setting where you look at patients where pathology increases the variability, the atlas will reach its limitations. And here's one of those examples. So what you see in green is a tumor. And in blue, we've reconstructed the corticospinal tract for this particular patient. And in red, you see where the atlas predicted the spinal tract to be. Now, if we were blindly trusting the atlas, then this could have quite. So it's always important to take into account the variability between us. Now, another question that came up is, if we are all different, does it matter? And I thought this was a quick question to ask. So in the early COVID days, I thought this would take a weekend. It took me a year in the end, because more studies looking at the variability in anatomy and the variability in cognitive profiles. And what you can see here is a couple of things. So number one, most of what we know about the function of the white matter, we know from clinical populations. So the majority come from neurology, neurosurgery, then psychiatry, and only 25% of the studies are conducted in healthy controls. What you can also see is that some of the tracts are superstars in one field, like the corticospinal tract in neurology, neurosurgery, and a lot less so in other tracts, other fields. And if you were to pick a tract to study, I would recommend choosing one down the list here, because there's still a lot of room for studies to conduct. Now, the other thing that we learned from this study is that, let me zoom in so you can see it, that there is no one tract, one function association. So I'm highlighting here the arcuate fasciculus, which is the language pathway, but you can see it also correlates with measures of memory, executive functions, auditory, attention, and motor functions. And we see this profile for every single tract that we looked at. So in the supplementary, there's 60 tracts in the brain. Every single one has this profile where you see that there's no one-to-one thing. Now, this is the healthy brain, but now the question is, does it matter that we're different in the presence of? Here's a study we did with acute stroke patients. They came into the clinic, got an MRI, got a neuropsychological assessment, including a language assessment, and we followed them up 6 months later. And what you see here is on the y-axis, their language recovery 6 months after. On the x-axis, you see the anatomy of the arcuate fasciculus in the healthy right hemisphere. And I'm highlighting 3 patients for you. You can see patient number 1 didn't really recover language well 6 months after the stroke. And when we look at the arcuate fasciculus in the right hemisphere, it's rather thin. Looking at patient number 2, that patient did somewhat better in terms of language recovery, and the arcuate is strong in the right hemisphere. Patient number 3 is in that blue bar, which according to the neuropsychological assessment actually means back to normal language, even though when you see the patient as a speech and language therapist, you still pick up on some deficits. And you see that that patient has a really thick, nice arcuate in the right hemisphere. Now, all this data is corrected for clinical demographical data. And if you just take the clinical demographical data, you can predict about 30 to 40% of the variance in recovery. By adding the variability in anatomy, you can nearly double that. So now you're reaching somewhere close to 59%. Now, another was that the first disconnection syndrome that was described was conduction aphasia. So these are patients that can articulate, they can understand. What they can't do is repeat what they heard. And that's because the arcuate was supposed to be disconnected. Now, I didn't see that in my stroke patients. And I walked around conferences, I asked experts, no one really saw it, despite 150 years of everyone repeating that this is the mechanism of conduction aphasia. So we changed our model to a different pathology, which is neurodegenerative in this case, primary progressive aphasia. And as I said, the first model was it's a disconnection between the frontal and the temporal lobe. With the emergence of CT scanning in the 80s and MRI later, we saw more and more patients that actually had a lesion to the inferior parietal lobe, but not necessarily the arcuate fasciculus. So now there's a body of literature that points towards the inferior parietal lobe rather than the white matter. So what we did is we looked at both. We looked at the white matter anatomy and the gray matter atrophy to see which one is actually driving these results. And what we can see is that for the arcuate fasciculus, the classical arcuate fasciculus, it's nearly a flat line. There's absolutely no correlation in the presentation of conduction aphasia and the anatomy of the arcuate. But we see that for the indirect segment, there is actually a correlation. Looking at the cortex, there was only one region that survived multiple comparison correction, and that's the inferior parietal lobe, this teal area here. So putting this evidence together, we, after 150 years of conduction aphasia being the arcuate fasciculus, and replaced the model with a lot more flexibility where a lesion can occur either at the cortical level or somewhere along the indirect segment to give the presentation of a conduction aphasia, but the flavoring will be slightly different. Now, in this exciting new time where we have AI to help us, so we took this work and tried to predict from the onset of a lesion where the journey will go long term. Now, if you just look for machine learning brain predictions, there is pages of pages in Google. But the idea fundamentally is very simple, and I'm trying to illustrate that here with something we're all familiar with. So we build our models by throwing as much data as we can at it. And here we have a model of climate change. So adding all the predictors, we know where the journey is going if we don't do anything, and we can somewhat predict where it might go if we do things. Now, one of the first things everyone is doing is like, okay, if that's the model, what does that mean for me? So if you look at the Netherlands in this particular instance, then our increase will be somewhere around 1%. So you make changes accordingly, trying to influence the model for the better. Now, that's very much what we do with our brain models as well. You have a model, personalized prediction, and then a tailored therapy for that. The only thing is that our models look different. So here's what one of our models looks like. So we took stroke lesion data and got the disconnection profile. So all the white matter connections affected by a given lesion. And we did this for over 1,300 patients, and we plotted them into the morpho space. And then what you can do is you can take an individual patient and bring that patient into the space, and then from there on, get a prediction of that personalized recovery trajectory on 86 neuropsychological behavioral assessments. A mental idea, and you can do that for various patients. So we see different lesions here, and it gives a very accurate prediction of outcome, 3 month, 6 month, and 1 year. The accuracy decreases over time as the model doesn't have the best data yet to predict long-term outcomes, simply because we stopped testing patients as good enough or not changing anymore. This was recently used by a team in London, UCL. They have tons of data. They collected stroke data for the last decade, and it's called Deplora's Predicting Language Outcome and Recovery After Stroke, headed by Kathy Price. And they basically use their data, which is out of sample, to the data that we used to train the model, and applied it to the model, and actually had a very accurate prediction for their own patients. Now, the good news is this model is available. So if you have data, all you need is the lesion of your patient. You go online and you can do the prediction for 86 cognitive and neuropsychological behavioral assessments for your individual patients. I want to leave you with this quote, which beautifully summarizes our thinking at this point, which is: the brain is so much more than its constituent cells. Each neuron in the brain connects with thousands of other neurons, but instead of a cacophony of connections, we have a synchronized symphony. Thank you very much.